Alex was talking about, none have any conservation status. So none of them are SARA listed or provincially listed. And as Maheshi was pointing out, with the decline in starling populations, none of the tick populations are in decline. So this is a bit different from the other ones. Now, we also get hitchhikers. This is when people, back in the olden days, you might remember like 16 months ago when people used to go traveling and they come back in here, they'd bring back extra visitors with them. And so these are the species that we would see coming off of people or people's pets after they come and visited. They have not established here. And there's only one that has the potential of establishing here, but each of them has a concern because they've been recorded to be hosts for a variety of fairly substantive diseases. Now, I know the Lone Star tick on the top there, it's a very easy one to identify, and the reason is that's one that's got a bit of headlines right now because the bite of a Lone Star tick in roughly about 5% of the cases can cause an issue with red meat allergy. And we have had 2 cases of that within the province, again, related to travel. So anyways, as you can see, we have, there's an additional 6 species here. And of those 6 species, 2, which is the Western black-legged tick and the castor bean tick, or the sheep tick from Europe, those are also capable of carrying Lyme. Most common ones we have here, well, what I've got here is I've got images of adult females, because that's what you're most likely to come across, or at least be able to readily identify. And these are them. You notice the scale on the bottom is in millimeters. The one on the left is called a groundhog tick. Now, see, features that we look at when we're identifying, features you should look at when you come across them, is you look at, oh, hang on, let me just go back here. This is what you call pushing too many buttons at the same time. So you have the features that we look at here is we have this cover on the back. It's called a scutum. It's sort of like a bit of a shield structure. And these are all females. So this is called a groundhog tick. You notice it's angular or subangular. It's a diamond in shape. It's got a pale surface. These are all unfed animals, and it's got club-shaped palps. So in other words, you're looking at the shape and the color of the scutum, the back, the abdomen, and the palps. This is the black-legged tick, unfed. I used to call them deer ticks, but again, there's issues with trying to get them a bit more specific to what we find. So here the scutum is perfectly circular. It is black. The legs aren't necessarily black, okay? Look at the palps. The palps are long and thin. So this is the only one that has very, very long, thin palps. There's a species that's appeared from the Far East. Is found in the States right now called the Longhorn Tick. We don't have that here. That also has them. But in our species suite, long palps here, and the abdomen is orange, sometimes a bit on the faded side, sometimes quite bright. So this is for unfed animals. This is the one that you're most likely to identify quite readily. This is a female dog tick. Some people call them wood ticks. Notice that if you look at the palps are short and stumpy, if I can get that area. Very short and stumpy as compared to the long, thin ones and the club-shaped ones there. Look at the scutum. Look at that shield structure. So to begin with, it's subangular, but not quite as regularly as with the groundhog tick, but it's got that cream flecking. Now, some other species also have those types of flecks on them. However, this is quite distinctive. Now, you'll notice also some other features along the back. These are diagnostic ones for later, but you can see the size. It's very, very large. It's quite angular in shape, very short palps. And this one here, which most people don't see very often, but it's probably the most common tick that we have here, and that's referred to as the rabbit tick. And it's got palps which are very similar in structure to what we have with the dog tick. They're short and stumpy, but look at the size. This is an adult female. The adult female is only about 1.5 millimeters in total length, so it's very, very small. And you'll tend to find them associated with snowshoe hare. So if you're out there right now going for a walk, as soon as we finish these sessions, and you do come across a tick and you bring it home with you, it's most likely going to be one of these 4. It could be one of the others, but probably not. Moving right along, misreported ones. This is the one that I referred to before, Lone Star tick. Now, if you find an adult female, it is easy as sin to identify. Notice on the back, you can see the scutum here. It's about the same color as the rest of the body, but you've got a very distinctive, almost like somebody went and dipped it into some whiteout. It does have long palps. Now, we do not have that one here, although it has appeared, it has expanded its range to southern Maine. So potentially within several years, they could appear. Every time we get a report, even now, we do a follow-up on it to see, and although we've had a number of reports, we have not found any evidence of them being here other than with hitchhikers. Now, basic tick anatomy. This is Sex 101, easy to identify when they are adults, and this is really important because the adult males only feed for very short periods of time in order to be able to maintain their fluid level. They do not stay attached for any length of time. The adult females, much more so. So what happens is the adult males also have a scutum, but you'll notice the scutum covers virtually the whole of the back. Okay, as opposed to a female. Now, these are both dog ticks or wood ticks, and you'll notice it only covers the front end. The reason is because the female adults will be feeding in order to get enough protein to be able to ripen the eggs, and of course, the abdomen will be expanding very substantially. The adult males do not. So therefore, it doesn't matter what species it is, it's the same pattern. So if this was a black-legged tick, this would be a circular scutum here. With the black-legged tick, male, that scutum would cover the whole thing. So if you see one that's an adult, that immediately you can sex them very readily like that, regardless of the species. And notice again, I mentioned before, the short and stumpy palps. We'll get back to how they use those palps later on. Now, this is the black-legged tick here. I have this illustration to show that they go through a series of stages in development, starting with an egg which hatches into a larva. The larva is very small. It's generally about 0.6, 0.7 of a millimeter in size. And notice that it has 6 legs. Okay, so this is, once you finish examining all of your lichens and your mosses that Alex is telling you about, take that hand lens with you and take a look. So you'll see 6 legs. Notice the long, thin palps in here. And this particular one, you can actually pick up a little bit of the gut pouches in the back. And gut pouches are important. We'll get into that in a few moments as well. Now, there is no transmission of any disease through any of the eggs of the species that we have here. So therefore, if you come across a larva, it's not an issue other than the fact that it'll tell you what species you have here. That species will feed on a host in order to be able to transform. He says, pushing the wrong button, it'll transform into a nymph. Now, notice how much larger the nymph appears as compared to the larva. The larva is the size of a poppy seed. Okay, the nymph is the size of a sesame seed or flax seed. Now, in here, you notice that once they've fed, they've transformed. They have now 8 legs. They're quite a bit larger. You can certainly see the pet pouches showing up here. But again, they've retained those long pouches. So go from larva to nymph. That nymph, if that larva has fed on an infected host, it will transmit that disease to the nymphal stage. Okay, so the nymph, if it has to transform into an adult, so it again requires a bud meal, and that bud meal means that if it has fed on an infected host, then it will then be able to transmit that disease to the next host. Once it's fed and it transforms, then they become adults. So here on the right, we have an adult female. On the left, we have an adult male. Now, notice the scutum here covers the whole of the back, and they're fairly easy to identify. Notice the palps here are a little bit more club-shaped. As a matter of fact, initially it was thought it could have been a different species. But when you see them, you'll see very distinctive white portion of the abdomen on the outside of the scutum. So easy to spot that. So again, if you see them, if you have one of those on you, again, not a significant concern. This is an adult female, so you can see the difference in the size going upwards. And the adult females can be, well, quite frankly, as long as you're as large as your imagination will let them be. So people will report them the size of grapes. People report them the size of swollen of raisins, and on and on. Anyways, looking at this species, this one here is an adult. So there is the circular scutum. There you have the orange abdomen, and you can just see faint traces of the 4 gut sacs. They call them cechi, and you've got those very fine palps. So you have those 3 stages, and it is the female who will be feeding upon a host in order to get the blood protein in order to ripen the eggs. So they're referred to as 3-host ticks. So going from an egg The eggs are laid usually this time of year. Those eggs will hatch through the summer, produce larvae. The larvae will overwinter. The larvae, you'll notice, the hosts will be feeding on very, very ground-dwelling animals, low ground-dwelling animals specifically. They'll overwinter to the following year. The following spring, you'll see nymphs. The nymphs will be available then to feed on larger potential hosts. And once they have fed, they will transform into adults by the fall, and they overwinter as adults. And of course, they're ready to lay eggs the following spring. They are active year round. So in other words, every season is tick season. Even though we don't see any more adults by midsummer, there are still potential ones there. So essentially, you need one host to go from a larva to a nymph, one host to go from a nymph to an adult, and one host to go from an adult female to produce eggs. So there's also a one-host tick. Now, if those of you who actually follow this guy by the name of Bob Bancroft, he did a talk on mainland moose several weeks back, and he was talking about a moose tick, which is sometimes also called a winter tick. Now, that's one which causes major mortalities in moose. It had only been occasionally seen here over the years. At the museum, we only had 3 records, and I talked with Lands and Forestry and their entomologist there, and he's only seen 2 in his career there as well over the years. However, they are a major problem in the Boston states, where essentially once the eggs are laid on an animal, the eggs will hatch, the larva stays on that animal, it will feed on that animal, transform into a nymph on that animal. That nymph will stay on that animal, feed on it, and transform into a male. So essentially, you can get individual moose. Generally, you see it in the younger ones with up to around 60,000 individual ticks on it, causing, and of course, they're all feeding on blood, so therefore, major problems. So it was seen here occasionally. I talked to the people at the vet college, the cooperative wildlife group, and they say now with the necropsies they've been doing of Nova Scotia moose, they've been encountering higher and higher numbers, especially from the cobequids in the last several years. So that's something that is changing. How do they get established? They have to be introduced. There was nothing here when the glaciers came through, and these guys need hosts. So looking as an example, the dog tick or the wood tick was introduced into Nova Scotia right at the county level here. The 3 counties join in 1896 with hunting dogs, and then essentially there was active monitoring. You notice this is 1940. That was the range of them in 1940. 10 years later, they'd expanded, so their core population was still in the initial place, and you see how much further they've gone out within those 10 years. Take another 10 years, they've expanded. Now, the ticks themselves haven't moved. They can maybe move several meters during the year. That's it. However, once they're on a host, that host disperses. And so really what we're looking is the dynamics of host dispersal. In this case, it would have been mostly deer, later on in the 60s, coyotes and foxes and the like. And you'll notice that up until the mid 80s, we basically were in Southwest Nova, but we started seeing something different in distribution where it wasn't strictly animal hosts. You notice that these are what in the military called communication corridors. They are now being found distributed along highway axes. And of course, the Chester Road or the Windsor Forks Road. And the last data that I had recorded, although there's more recent data, you can see by the mid 2004, 2005, they had now expanded substantially, very much by us. So we're the ones that started moving them. And you notice little red dots here. This is places where we've confirmed they are and established. These are roadside picnic parks where people take their dogs out to give them a quick run because they need a pee after driving all over the whole province. Now, since this time, actually up to 2020, all of the mainland now has a good healthy population of dog ticks. And this is the reason why, is because we've got lots of good habitat available. As a matter of fact, good place to look for ticks right now. I'm not supposed to mention specific commercial places, but if you go to a Mastodon Ridge, back in behind the Tim Hortons, they've got a lovely 1.5-acre dog run park where up until the close down with the tourism coming through, they figure between there and Mastodon, those 2 places, 60% of the tourists visiting Nova Scotia would stop at those 2 places. So anyways, tall grass, watch out. Once they're established, how do they spread? By a host. It could be a mammal host, it could be a bird host. How do we determine where they are? We can actually actively sample, which essentially is for all you men out there, we can get away with this once, which is take your best sheets, cut them into 50 centimeter by 1 meter strips, put them on a piece of dowels, and drag them behind you, going through the forest. The ticks are sitting on vegetation, on structure, waiting for a host to come by. They get knocked off, and then if you say, for example, these are 1 meter by 50 centimeters, you do a 200-meter transect, you've just gone through 100 square meters of habitat, and you can quantify how many ticks you have. The other way is passive reports, and every time, although we tend to grumble and grizzle about taking our pets to the vets and getting charged so much, well, they do a lot more than just looking at the specific problem. They're also looking for ectoparasites, and they've been a very, very significant source of information pertaining to distributions. Now, black-legged ticks introduced in the 80s. How did they get here? Migratory birds. We're fairly sure of that, and this is the reason why. That by 2006, when we plotted out all the places where we confirmed the ticks had been found, these are not established, these are found. You notice that it's like a scatter plot right across. Now, we have actively looked for them in 9 counties by that point, and the distribution was still fairly tight as to where you'd find them. So in all likelihood, introduced by birds, and you can pick up, certainly even on the Northumberland shore, you're starting to pick up a pattern, perhaps a jump-off pattern to go further north. Now, when we start looking at the places where they were confirmed back early on, we put a 50-kilometer radius around each of the spots. This is distributions based on Department of Health and Wellness back then. When we overlaid where we had specimens submitted to the museum, you can see we had very good congruence here. So the Halifax, the Bedford area, Lunenburg area, Maiders Cove, Shelburne, Yarmouth, and on. And then we had a couple that there were no reports from within health, and certainly none in along here, although we were getting very large numbers from Amherst all the way back over to Tatamagouche. Now, with time, so what they've done is they've changed, they give you a risk map. So this is the original format that they started using, and I think this was 4 years back. And essentially, if you go on their website, they indicate the areas of risk pertaining to Lyme. Lower risk means that the ticks may have been discovered there, but there's no guarantee that they're established and there's no disease. Moderate means the ticks are established, no confirmation of disease, and higher means that the ticks are established and Lyme is present. So with time, you'll notice that that has changed, and this is the current distribution. So essentially, Guysborough County is still not confirmed through as Lyme being established through there, and we certainly have Cape Breton County. So they have been slowly expanding through the province. Now, the interesting thing is that when you look at the black-legged ticks, which is the only species that can carry or is able to carry Lyme, and transmit it to another host, it depends where you are. In some populations, only about 25% of the ticks carry the bacterium. In other places, 40% of the ticks carry the bacterium, and it doesn't change with time. So in areas around Bedford Basin, for example, it's running around 25%. In areas east of Picton and Glasgow, it's running 40%, and there's a reason for that, we think. Anyway, so let's get on to some natural history here, and the following several images come from Krista Melville, who is with the Halifax Field Naturalist, and I think she's with the National Research Council. Amazing photographs. This is a dog tick or a wood tick, and you'll notice it's attached. It's sitting on a piece of grass. It's got 8 legs. 2 are holding onto the grass. It's pointing downhill. It's pointing down the stem, okay? Notice the big palps there. The other feet are out because you have 2 things. You have the sensory organs, which we'll get into in a second, and also you have the ability then, if the feet are out, in order to be able to snag something when it comes by. So that was a female, this is a male. You notice the same thing, holding on a little bit tighter, but you notice certainly the forelegs are out there. Remember I mentioned the scutum, notice how the scutum carefully covers the whole back there. And the other image here, so this is a female, also just with her arms out to the front. And they refer to this feature as questing. So essentially they're sniffing the air for the possibility of a host. Now, if you look at black-legged ticks, they also quest, But if you look carefully, they're questing by pointing upwards, off the tips of the grass or off the shrubs. So it's quite it's a bit of a behavioral difference between the two species. Again, this is a female. Now this gets us to what are they doing when they're questing? Well, getting back to this image, let's take a peek at these feet. And with those feet, essentially they have a claw on the end, and that allows them to grab on. So if you have one that's walking along you, it's probably using its claws. If you try to grab it, it may grab onto the skin with the claws, but if you let go, it will let go and keep climbing through. Second thing is it's got a pad on the base of the claw, very much like a fly or a spider, which allows it to stick to smooth surfaces. So whether it's smooth leaves, skin, or even glass, it's quite able to do that. So that's its holding on mechanism. However, this is the important part. This is a structure, a pit near the tip of the leg called a Haller's organ. And when you look on it, it has a suite of different kinds of sensory hairs. These sensory hairs are used for detecting signals. They detect chemical signals and physical signals, those specific cues. The chemical cues they seem to target on is very much like mosquitoes, black flies, and the horse flies, deer flies, essentially looking for carbon dioxide because that is a sign of respiration. They're looking for ammonia. Again, that's a sign of physiological of metabolism, and they're also looking for lactic acid, the sign of muscle activity. So again, it's hints that there's something there that potentially has blood. They're able to cue for temperature, and I suspect that might actually be infrared. So the whole point is that, remember I mentioned they were questing with those legs. You see one sitting there, just watch it. It'll be moving it'll be swinging around with those front legs, tasting the air or testing the air for possible hosts coming through. And you can actually play with your ticks by putting them down on a piece of paper and then put a finger in to one side of it, and it'll orient towards your finger, and then start moving towards it. Shift your finger, it'll orient towards it. It's not because they can see, because they don't have a head, they do not have eyes. So it's strictly a chemical thing. Now, possible hosts, basically any warm-blooded animal that we have floating around here. So it doesn't matter at the earlier stages whether it's something like our squirrels and chipmunks. This is probably the most significant host for most ticks, and certainly very significant for black-legged ticks. So whether it's the deer mouse or whether it's the white-footed mouse, which we have, between the two, we have good coverage across the province. Those are the most common ones for maintaining the populations. They're low to the ground, so the ticks. Oh, by the way, Alex, here's what kind of moss is that on? Okay, you'll have to answer that later on. Okay. Anyways, but sometimes you do find them on less common hosts, and although we see things, oh my gosh, we got them on the dog, or we got them on the cat, or we got them on the person, those are not very common hosts because they're not in that same habitat picking up the same numbers. Now, we have a farm. We actually have dogs like this. These ones have very few tick exposure because it's a very long and very thick fur, very little exposed skin except right around the muzzle and the eyes and a little bit at the ears. Sheep, however, their bellies generally tend to be almost completely bare, a little bit of wool on the brisket. Sorry, I'm a sheep farmer, so we sell them for meat. So very little meat on, very little hair, very little wool on there, and very little hair, and totally bare skin, very thin. So they should be able to punch their way right through, but they don't. And we suspect it has something to do with what's called wool fat or lanolin. It's acidic, it's greasy. It may have some features that may be useful with respect to trying to get rid of them. Okay, I seem to be missing some slides here. Okay, so we're going to have to just quickly going through here. This is a schematic of a tick, a black-legged tick. Specifically, I mentioned they have these pouches. When they feed, they actually swallow in a peristaltic manner, very much like we do, even though they don't have a pharynx like we do. The fluid then comes in through the mouth parts, and then it starts to fill up these pouches. So it's got two pouches here, it's got two more pouches there. And as they fill, then you start getting then they start swelling up, and that's when you start noticing that you've got an engorged tick on you. But as they're feeding, as that blood is consumed, they consume more than they need, and then they can use all the bits and pieces of. They do not urinate, so what they do is the fluids go through the gut wall and then through the hemocoel into the saliva, and it goes out. And a lot of stuff goes out through the saliva because when they're feeding on you, they have to put an analgesic in because you don't know they've bitten you and they don't know they're sucking at you. They put an anticoagulant in because they're going to be attached for up to around 72 hours at least. So they don't want the blood to clot. They put an anti-inflammatory in there because they don't want that immediate response. They're blocking off flow of food. So, of course, if they have disease and that disease can be transmitted, that disease may travel through the gut wall into the hemocoel and into those salivary glands. So I'm not quite sure which slides disappeared here, so bear with me as we're going along. Why are they important? Because they can carry diseases that people can encounter. And as a matter of fact, most of these are referred to as zoonotic diseases because several different species may have them. And you'll notice that some of the species, this is when they were first discovered, Lyme disease is a very recent discovery, basically within the last 40 years or so. And it is increasing in both distribution. Here we go. Okay, this is getting onto the feeding part. So essentially, this is the basic anatomy. Those are the things. Now, why does when we go back into here, they feed the excess water goes through the wall and they start concentrating the blood. Then is used in the case of Lyme to also help culture the bacteria. The water and some of the bacteria go through into the salivary glands and they get injected back into the skin. Now, this is a mammalian cross-section of a mammalian intestinal tract, and you'll notice, but it's somewhat similar to what we would expect. You have a hole in the middle for the food to pass. It's called the lumen. You've got these villi or microvilli. They call them. Come to think of it, they call it tunica mucosa. So essentially, a very fine single-celled layer through here, and then of course you'll have a series of muscular walls going beyond that. Now, going into this is where at the bottom this would be the middle of the digestive system. Here you have the muscular portion, and here you have the villi. On the surface of it, we have a whole suite of things. They could be peptides, they could be bacteria. There are a number of proteins that are contained within the mucus in there. So what happens is if you have a bacterium within the gut lumen, which is here, the bacterium then will pass through these surfaces if it can. It goes through the villi, gets absorbed, it carries on to carry on into the rest of the body. However, in most species, the bacteria do not make it through. You can have a wood tick, or you can have a squirrel tick, or a groundhog tick, whatever, feed on a Lyme-infected host, but the Lyme bacteria will not make it through this particular immune system. So essentially, they block it. Now, getting back to where I said that 25% or 40% of the ticks may carry Lyme, well, there's a variation in the microfauna and the microflora that you find within the gut. And it's very stable within most populations. So we suspect what's happening is that those 25% do not have the ability to inhibit the bacteria, or 75% do. So essentially, it's the tick's own immune system is acting to reduce the chance of the bacteria from getting through. And there's some recent work being done out of Boston right now with respect to trying to enhance that immune system on the ticks, therefore to make them less capable of passing on. Now, quick set of images here. This is an adult female black-legged tick, and here you can just see the pouches. So you can actually detect how long they've been feeding. This animal has not been feeding, so you can just barely see the pouches. As they feed more, the pouches start to expand, start to show up, and by the time you see something like this is usually about 36 to 48 hours. These animals have been, if you see that, you know they've been patched that long, but also then by this point, you now are acting as a culture medium for a variety of bacteria, including Lyme. Notice that although it looks like it's embedded, there's a little bit of inflammation here. The only part that goes into your body is the tube by which they suck. So you say, "Oh my God, it was buried in the skin." Well, no, actually, only part of it was. And this is a gorgeous shot of an adult female. She is fully fed, and she has pulled herself out, and you can see the intact mouth parts here. There's a perfectly round shield on that scutum. So you get an indication as to how much that is actually swollen. But notice back in here, a little structure back in here. And this is important from the point of view of how you can control them. How do you remove them? On the back, they have these two plates. Now, if you're looking at insects, insects don't have lungs. They have a series of tubes that go through the body, and air passively, more or less passively, moves through the system, and you get oxygen exchange and respiratory byproducts removed. Within ticks, because especially with the females, they're going to expand very, very dramatically. They don't have a series of tubes per segment because they don't have segments, but they do have several hundred, if not some up to, depending on speed, several thousand tubes. And all those tubes come out in these little features towards the back and behind the legs. And here you can see the individual tubes that are lined up in there, and that allows it to keep on breathing. And also those tubes, and it's only the ones to the hind that get stretched for purposes of respiration. Anyways, the point being that you don't see dead ticks lying in heavy dew, and the reason is in around the entrance to each of these holes, you have a bit of oil, and that oil stops water from getting in. So in actual fact, if you wish to quickly kill a tick, if you just apply a little bit of water with a little bit of detergent on it, any kind of a surfactant, it breaks the oil down and they drown immediately. It's very, very quick and painless to us, of course. Now, do not use Vaseline, do not use oil, do not use matches. The reason is because if you use those, yeah, you'll basically block the breathing tubes up, and they cannot get air exchange. However, at the same time, getting back to this image, you'll notice that you've now basically caused it great distress. Those tubes will be coming out back here. And it's just like you, if you get into great distress, the chances are fairly good that you will empty your gut contents. And that is exactly what they do. If you do that, they will empty all of the gut contents back out through the skin and into where you don't necessarily want them back. So it doesn't matter what they had. You're going to enhance the injection. So do not use those things. How long do they stay attached? Blacklegged ticks, very short length of time, 72 hours. So that's basically up to 3 days. No, they weren't on you for 3 to 4 weeks. If you had some earlier on, then chances are you've had several groups of them coming through. However, to stay attached, how do they do that without falling off? And essentially what they do is, I mentioned that the males only stay attached for short periods of time just to feed, but the females have to stay attached the minimum of 3 weeks. They don't have muscles holding them in place. So what they do is they cement in place. So this A is a schematic of a wood tick or a dog tick. B is a schematic of a black-legged tick. They produce a biocement. This is the skin. These are the palps, and they produce that biocement within about 12 to 24 hours, and they cement in place. So with the dog ticks or wood ticks, they cement on the surface of the skin. With the black-legged ticks, they secrete it in along the feeding tube, so they're well within the skin. So if you pull one of these things off, generally speaking, you'll come off with a hunk of skin, generally about that much skin in the first place. With black-legged ticks, they're very, very difficult to get off. You actually have to pry them off. Or if you're like me, you're cheap and you can actually make a fine slip knot and using a piece of thread and pull them off. So you can there's a variety of different kinds of tick removers. Essentially, they're small pry bars. They're designed that you basically pry them off the skin without squeezing the guts, because you squeeze the guts, gut contents will go back inside you. Where do you find them? This is, by the way, this was approved for the elementary schools in the system, so I hope nobody takes offense. This is your basic body. These are the places with respect to black-legged ticks specifically, because there's a bigger concern. You can find them anywhere on the body. However, they do have preferences. And essentially, you're looking at places where there is less chance of getting rubbed off. So this is what our little boy, who's now 41 years old, used to call the arm bow. So in the elbows, armpits, under the breast line, the groin, and of course, in between the thighs, the back of the knees, and butt crack. So I've been looking, by the way, for good images of Lyme infestation, and you get the pictures available on of different places of different kinds of rashes. So here's one example. So you get a rash without the bullseye rash. I have yet to find a single good shot, because butt cracks are one of the preferred places, of a bullseye rash or any rash on the butt crack. And if you do come across them, please pass them to Bob Bancroft and he'll forward it to me so I can include it. So what you do is you're looking for, if you know there was a bite in around the side of the bite, you may get a rash. The rash would be, generally speaking, 45 millimeters, let's say 2 inches or larger. Anything larger than that, doesn't matter whether it's a bullseye or not, that immediately is suggested. This is more characteristic of the bullseye type rash. So again, if you have those kinds of symptoms, and you don't know what the tick was, that's an 811 question, or consult with your family physician, or certainly do a follow-up with that. Now, so this is the species we have, the black-legged tick. It's the only one here that can carry the Lyme. And by the way, again, there's those pouches showing up in there nicely. They are cyclics. They're available year through. You'll find them generally on smaller hosts earlier on in their developmental stage. The larvae on essentially small rodents and birds, nymphs, much larger animals, and essentially the same thing with the adults. Where do you find them? Great habitat. Essentially, they're looking for that structure. Remember the picture from that Krista had there? So it could be grasses, it could be ferns, it could be any kind of a vegetation, which allows them to climb up and wait for a host to come by. The host will dislodge them, they'll grab on and start climbing. If you like fishing, then again, this is looking at alders. Again, people report having seen them falling out of trees. It probably does happen, and the reason is because although we know a lot about ticks, we also know that ticks can't read, so they don't know what we know about them. But the point is, if you like fishing for these neat spots in here, these are good potential sites like that. A special case, you probably recognize this from, what, 13 years ago? I guess it'd be about that. So it's a fire back in Spryfield, Harrietsfield, York Redoubt, that area in there. And essentially after the fire went through, you'll notice that the ridges, the upper areas had been basically burnt off. You got very little vegetation left at all on the surfaces there. But in between, of course, you had a lot still growing. So people said, oh, good, that got rid of the ticks. Well, within about 10 to 12 days after the burn, now this is the one from down near Keji, you started seeing ferns growing back up. This was several years later on, and guess what you found on those ferns? So essentially, as long as they had a refugium or point of refuge during those fires, then they're going to go up the nearest and the first structure, which is there, which are the ferns. So essentially, they're designed to be able to make more copies of themselves. Other places you can find them, you got, I've seen some gardens, not quite like that around here, but essentially what have we got in here? Although it's ornamental, from the tick's point of view, loss of vertical structure. Gardens, tremendous, lots of vertical structure, okay? And flower gardens, even much more so. So even if you're gardening, please take a peek when you come out and check through. So this was really, really funny, because when they changed the law allowing you to grow these funny looking hybrid tomatoes, and I did a talk at one of the garden clubs, all of the people, all of the old gray hairs, blue hairs, and no hairs, as I fit in that, Bob and I fit in that category, immediately recognized what these were. But when I gave this talk at a couple of universities, most of the kids had no idea what they were. It was actually a bit on the humorous side. There's some nice shots of lovely places to go and enjoy the great outdoors. This was just posted 2 days ago. Not recommended. It'd be perfect tick habitat. So again, you're going to do that. I suggest there's an issue with the tick check. Are there risks available with because of ticks? The answer is yes, but the risk is manageable. How do you do it? Simple. Before you go in the field, then you can use have a number of items that are registered for use. The DEET-based products are one. Keradin is another one that's now licensed for use. And there's a permethrin that's available to be used that you can purchase clothes that are impregnated with it. So those are quite effective. Now, call them repellents. They don't repel, but they basically block their ability to be able to detect the signals coming up there. Wear long sleeves, shirts, pants, shoes, tuck pant legs in. Somebody asked at a phone in, what about rubber boots, 10-inch high rubber boots? I said, well, they work really well up to 10 inches, but then you're up above that. So again, Once they get on you, just as with vegetation, they start climbing until they find some bare skin. So again, tuck your socks into your boot on the outside of your boots rather, and have everything else tucked and nothing exposed, and you're good. And then do a tick check when you come in. And it's very straightforward. Start at the top of the body, work your way down. And this is important because actually in the last 3 years I had at the museum, the majority of the species that we're coming across were being reported from the head, generally associated with the ears. This is a female dog tick. Their preference actually is for the scalp line. So for people like Bob and me, that I think if we were to do a groove through the halo of hair on the back there, they'll keep on going for the scalp line, bypass that, carry on over the forehead, and then we won't have them again. However, the point is that checking through every possible place that you can, tick check is important. If you some places you just can't see, so you should have a friend check them. For some places that are a little bit more sensitive, well, let's just suggest a close friend will be preferable on that. So doing that tick check, and then the following day, whether you do it during a shower or whatever, do another one in the morning, okay? Because the ticks have been climbing, they could be on your clothes, and they haven't, it takes them usually about 12 to 24 hours to get to a feeding position and attach, quite readily to remove. And once they start feeding, it usually takes, from the point of view of Lyme, 24 to 36 hours for them to be able to transmit. So again, this is it's not a rush thing, but a quick tick check always does good. And I got several calls from people, this is years back, saying, our grandkids are coming from Ontario and we got ticks here. What do we do? I said, well, turn it into a contest. The kid who finds the largest number of ticks gets a toonie. And I guarantee you the kids will find them all as soon as you give them an incentive like that. So demystify it. So yes, there can be issues, there can be dangers, but if you follow basic precautions, you can get around the thing. So proper equipment and proper procedures, you can virtually eliminate that risk. So are they risks? Sure, but it's a manageable one. Anyways, many thanks. And certainly nowadays, please stay safe. I don't know how we are for time because I don't have a clock. I can't see that at the same time. So we can either have questions now or later, or you can just pass them on to pass them on, and I can deal by email. We're there.
Thank you, Andrew. We are already at time, so we'll have to skip questions, but we will make sure that any questions you ask tonight will get answered. We'll give you an email, and we'll also post some links here in the chat. So there is a link posted to an example remover tool if you want to get some of those for home. We'll also post some links to things like eTick, places that you can submit ticks to if you're interested in that kind of thing. We'll make sure that you have Andrew's email. Ron, you're up next for us. I wonder if you could spend maybe no more than 20 minutes, because I don't want to be too late for folks tonight, talking to us about herps.
Okay. Okay.
Just
How
a second.
do I share